very pleased to have Stephen Quake here today from Stanford University. He's given us two beautiful public lectures, and today we'll hear, I think, more details in the same title, Precision Measurement in Biology.
Thanks. Today I thought I'd tell you a story about something we've been working on for several years now on how to do single-cell gene expression. Much of the data is unpublished, and I hope you'll enjoy the story. So I talked yesterday about these measurement techniques that are changing biology right now, and I'll walk through a bunch of these examples. I won't dwell on it today, but this area of single-cell genomics has been sort of something of tremendous fascination for me. And I talked a little bit yesterday about how to sequence the genome from a single cell, and today I'm going to talk about how to measure gene expression. And I'll give you a few examples in a couple of slides, but one of the very first things that one realizes is that there's an enormous amount of variability in gene expression between individual cells, even if they're nominally identical, if they're clonally related, and you're looking at boring genes, you see a huge amount of variation. Some of this is sort of systematic variation, and it derives from the cell's fate choices, its phenotype, whether it's a heart cell or a lung cell, and so that's sort of one source of variation. And some of it is statistical. Let's do the stochastic properties of the gene expression machinery. And I'm going to talk about both of these aspects and try and dissect them a little bit for you. So we're going to continue to use these plumbing toys that I talked about yesterday. These are some more examples of some of the chips we've made. And here's early one that we used for doing single-cell RNA preparation for single-cell gene expression analysis. And there's been a series of papers over the years where we've kind of progressively refined this, and we've got it working pretty well now. So here are some experiments that were taken with that first chip by Josh Marcus, who's a student in my group who has a clone, got a twin brother, identical twin brother. I didn't have the brother as a student, but this sort of illustrates what happens when you're looking at cells that are nominally the same genome, just one division, a couple divisions away from each other. And this is histograms of gene expression for, in this case, beta-actin, so the log of the gene expression level. And you see the distribution in log space is pretty well fit by a Gaussian. Okay? And same is true for GAPDH, another sort of boring housekeeping gene, shouldn't be doing anything exciting, and PGK. And so, if these logs are normally distributed, that says the RNA transcripts have an apparent log-normal distribution, which is quite broad, right? And this is something that not unique to our observations. There's quite a bit of literature even before our work on this, and everyone who looks at it sees more or less the same thing. No matter how you do the measurement, you see quite broad distributions.
Could you say just a word about the mechanism that would produce log-normal distribution?
I'm going to give you more than a word about that if you hang on for a little bit. I'll give you more than a word. Here's another thing to sort of set the stage. So this is these are single-cell gene expression experiments done by my then student Luigi Warren. These are primary cells from the blood of mice, and they've been sorted using surface markers. So by staining the surface of the cells with antibodies, we more or less know what type of cell they are. These are different flavors from the hematopoietic system. He looked at gene expression of 6 genes per cell. Let's just focus on the left figure, which is 3 of the genes. And so each axis is a different gene, CD45, RPL15, and beta-actin. And each data point is a different single cell. And so the point in 3 space indicates what are the expression levels of those 3 genes for that cell. And the cells have all been color-coded according to their identity that was measured by these surface markers. And so what you see kind of amazingly is even though these 3 genes are very boring genes that aren't supposed to tell you much about identity, they do a pretty good job of separating the cells according to their cell type. And so the notion is that, the sort of intuitive notion is that you can actually sort of measure cell type if you look at gene expression of a large enough number of genes, and it doesn't matter which genes you pick. That's sort of the hypothesis here. You look at enough genes, different cell types will have different expression levels of even boring genes, such that it sort of partitions them in gene expression space. And so we try to exploit that to try to dissect complex tissues, and that'll be sort of a big part of the second half of the talk. So let's return to this question of noise and statistical or stochastic variation. These questions have been really intensely investigated over the last decade by a bunch of people. Some of the names associated with it are Mike Elowitz, Ido Golding, and Sunny Shi, and they've mostly focused on bacteria. And much less is known about eukaryotes. There's a bit of a literature, but it's sort of a small one. And we decided to kind of dive in. And one of the things I'll just mention is this curious result by Sunny that in their bacterial measurements, they looked at the correlation between RNA and protein within single cells of the same gene, and they found that correlation was 0. Kind of a striking result. And it got a lot of people scratching their heads. And so I'm going to revisit this question here in eukaryotes and describe to you a little model that explains both our results and Sunny's, I think, in a pretty general way. And then I'll return this question with these log-normal distributions. So the simplest possible model for gene expression that a physicist can write down looks something like this. DNA makes RNA, makes protein at certain rates, and both the protein and the RNA get digested with certain rates and go away. And so you can write down a master equation for this model and compute all the moments, the variables. Many people have done it. It's pretty straightforward. And you can get some approximate solutions for the distributions of quantities. And so the, one sort of approximate solution for the protein distribution is this gamma function here, which is true in certain limits. And it's one that's favored. This distribution is something that's favored by Sunny, for example, in analyzing his data. And the RNA distribution here looks just like telegraph noise. And it's reasonable for bacteria, but it's not true for eukaryotes, as you saw from our distribution. So what's the next most simple model you can write down? Well, you can imagine that the gene isn't always on, that there's some promoter there that's fluctuating between on and off, either because a transcription factor is binding or chromatin is opening or closing or something like that. RNA polymerase is binding, falling off. Lots of things like that. So it's cycling between an active and an inactive state. So if you add that to the model where the DNA is randomly going between active and inactive, everything else is the same, you can do the same sort of thing. Write down the master equation, compute the moments, and now the RNA distribution in some limits is described by this gamma function. That's turns out to be not a bad approximation. I'm going to show you some examples of the measurements we've made and how we fit it both to the gamma function and a log normal, and how a little later how we've computed some of the other moments of this model and compare them to our data. This is sort of the picture you want to have in mind. And the other thing you want to bear in mind about this is you better darn well hope that it's wrong, okay? That we really want to falsify this model with our measurements because it neglects a really important part of biology, which is sort of programmatic control of the cell, right? We know it's not all driven by random fluctuations, that there's sort of deterministic programs that go on when the cells try to do things. And none of that is included in these models. This is just a simple model of fluctuations about some steady state. And so while it may describe some genes, I'd be shocked and very disappointed if this described everything we measured. So one of the tools we've used for the measurements I'm going to show you here are these microfluidic chips. And we, at one point, designed a chip that looked like a matrix, had a bunch of rows and columns, and everywhere they intersected was a different sort of reactor to do measurements. Now this lets you do kind of nice combinatoric experiments. And so our initial one was 20 by 20. This was designed by Jian Liu, who was a student. 20 by 20, which gave 400 reactions with only 40 pipetting steps. That was a nice economy of scale. And it would use it for PCR. And then Fluidigm built a commercial version that was 48 by 48, and then they made one that was 96 by 96. So you can do very large numbers of gene expression measurements or genotype measurements per chip. And the way we use these is we put the lysates in the rows and the PCR primers in the columns, so we can look at 48 or 96 genes per cell and 48 or 96 cells at a time. And that lets us get sort of a pretty reasonable amount of data. And we've worked pretty hard to understand the measurement noise in this system. and I'll show you a couple of examples. Fluidigm did a lot of work. They published papers showing that the measurement noise is less than you get in microliter volume PCR machines. Repeatability is a bit better and so forth. And we focused on measurements and these single cell measurements to try and understand what the noise is there. And this is some data taken by Tomer Kaliski, whose work you'll be seeing for the next several minutes, who was a postdoc in the group, now a faculty member at Bar-Ilan. And this is work we did with Bob Tejan at HHMI, looking at embryonic stem cells, and his postdoc was converting them into neural progenitors, into neurons. And this is kind of every data point here is a different gene measured over many different single cells. This here is the mean expression level in log coordinates. This is the standard deviation of the gene expression across many cells, again, in log coordinates. So the data points are the data from the cells, and the pluses here are data from control experiments with purified RNA that's been diluted down to more or less single cell levels. So this is an estimate of the technical noise in the measurement, and you can see that it's less than the biological variation in the measurement. That makes sense? Yep. And that's all I'll say about that. Here's another way to look at that in a different cell type. These are now human cell line HCT116. And this is now titrations of the total RNA. This is the sort of measured expression level. This is the amount of mRNA put in the experiment. Titrates very nicely. And this band right here is the range of expression levels measured in single cells. So this shows that we're well in the dynamic range, and that the sort of variance from the single cells is larger than the variance from the repeated measurements. And right below the graph is the actual data from the single cells. Okay? And these are for several different genes. We've done this for many of them. And so we feel like we've got a pretty good handle on where the technical noise in the measurement, how repeatable it is, and that the biological variation is exceeding the technical variation in all the cases we're interested in here. So then we looked at a large number of these HCT116 cells and looked at a large number of genes and fit the distributions both to log-normal and to gamma distributions. This is, and we also, I should say, besides looking at mRNA distributions, looked at a few protein distributions. So we cloned GFP, a fluorescent protein, into this cell line, so we could measure the RNA from the GFP and the protein level by looking at the fluorescence. And we also used antibodies to stain two surface markers, so we could measure the protein levels of those membrane proteins with, and as well as their corresponding mRNA levels. And here are some examples. So these are ones that were well fit by Gaussian functions, or sort of log-normal distributions. So CT is the log of the gene expression value, and so reasonably Gaussian fits. These here are ones that were not as well fit by Gaussian, but were better fit by the gamma distribution. You can see there's a bit of skew on the side here. That's sort of the reason for the poor fit of the normal distribution. And these are the p-values for the fits, where across a large number of genes, and RNA here and protein here, these squares are the p-values for log-normal, and the triangles for the gamma distribution. And so if the fits are systematically wrong, you'd expect a p-value well below this line. And so the p-values that are above the line are saying that, well, these fits are, the data are consistent with those distributions. And by and large, there's not that much of a difference between log-normal and gamma. Gamma's a little better in some cases, but generally speaking, I think I'm happy with both of them. And as you'll see, there are some good reasons to use log-normal. It's a lot simpler to interpret as a rule of thumb. Now, I mentioned we'd expected that some genes we'd hope would not be consistent with the model, and indeed, some just had fits that were clearly not even close to either of those two distributions. Here's an example of some of those. The distributions tend to be bimodal and even broader than log-normal or gamma. And these are genes, interesting enough, that tend to be involved in cell division. So we think these cells were in the process of of dividing, and you're seeing sort of separation to different sort of deterministic states. And there's strong correlations between these genes. And so if you take this one, top 2A, and you say, here's the distribution, and I'm going to draw a line right in the middle, and I'll color the ones on the right of the line red and the ones to the left of the line black, and call those my high and low expressors. And now I'll go through and I'll look in other genes within all these red cells and other genes with all these black cells. And so here's A and LN. And you can see that in all the cells that were expressing top 2A at a high level, they're also expressing A and LN at a high level. And they're also expressing BRC5 at a high level. And likewise, for the ones that have low expression, those correlations carry through. Same thing with STMN1, but not the case for every gene. So here's a couple where they weren't correlated at all with the top 2A expression level. And so there's interesting structure in the correlations between gene expression levels. And here's sort of the correlation matrix. The strongest bit right here, these are the ones I just pointed out, ANLN, top 2A, STMN1, and BRC5. And there's other sort of smaller ones that are cropping up and look sort of interesting. And it's sort of another level at which one realizes this model is neglecting something important. There's the fact that gene expression is correlated, especially when you go on programs like mitosis. So I mentioned you can compute some of the moments. One of the interesting ones is this correlation between RNA and protein level. And you can sort of compute that, and the model predicts something, and it depends on the parameters in an interesting way. Sort of the most one I'll focus on here is this one right here, which is essentially the mRNA lifetime, measured in units of the protein lifetime. And as you tune this parameter, you can see that you go from zero correlation to high correlation, and sort of an obvious interpretation. If the mRNA has a very short lifetime, it's not going to stick around long enough to be correlated with the protein expression level. And that's, in fact, why in Sonny's results, he was getting 0, because in bacteria, the mRNA lifetimes tend to be very short, whereas in eukaryotic cells, they can be much longer. And so you might have a completely, be in a different range here in eukaryotes and expect to see some correlation. And these are simulations for various choices of model parameters that show that you sort of can get non-trivial correlations. And the actual data shows that for the things we looked at, there is in fact a pretty strong correlation. This is the protein level for GFP. This is the mRNA level. Each point is a different single cell. And you can see there's a very strong correlation. And we have, in fact, measured RNA levels not just for GFP, of course, but for all those other genes that are in the panel. So we can compute the correlation between RNA of any of these genes and the GFP protein level. And those are all very low, as you'd expect. And the one outlier here is the actual transcript that's associated with the gene. That all makes sense. Same is true for CD49, which is one of the surface markers, non-trivial correlation coefficient, and the transcript for that gene is most highly correlated with the protein from that gene, much higher than any other transcripts are. That all sort of makes sense. And so, you can sleep easy at night. You don't have to stay up worrying that all these RNA measurements are useless because they're not correlated with protein levels. In fact, in many, many situations they are. And this sort of bacterial case is a bit of an outlier. All right, let's come back to log-normal distributions. So, as you know, the central limit theorem says that you take the sum of random variables with almost arbitrary distributions, you end up with a normal distribution. Corollary to that is if you multiply random variables together, the distribution of the product tends to be log-normal. You simply take the log of the product, apply the central limit theorem, and there you go. And so that sort of makes sense here. You think about how RNA and protein is made. There's a bunch of rates that have to happen in the cell, and all those rate constants are fluctuating randomly, and so you're actually multiplying a bunch of random variables together. So it makes sort of intuitive sense that you'd end up with a log-normal distribution in a very sort of general way. Now, log-normal distributions have this interesting property that in linear space they're skewed, and so if the log of the variable is normally distributed, the variable itself is going to have this sort of skew to the distribution. And it turns out that the median, the mode, and the mean are all different from each other in log-normal distributions in real space. And the sort of thing that you would think about as the central value, which is e to the mu, so mu is the offset in the log space. Think about that as a central value. That turns out to be the median in linear space. And the mean is actually displaced by sigma squared. Okay? And so the width of the distribution comes into the mean value, and that's sort of a very non-intuitive way to think about things. And so, and maybe a non-representative value. And so if you ask, what's the typical cell's RNA level, I would argue that the mean is the wrong thing to think about because it's convolving the central value with the width of the distribution. And how do you measure the typical cell's expression value? Well, without the ability to do single cell experiments, what you do is you do a bulk experiment on 1 million cells, measure some RNA level, divide it by 1 million, and you're left with the mean. But what you're really measuring is this combination of the central value and the width of the distribution. If you make single cell measurements, you can measure the whole distribution and calculate things like the median, the central value, and you can get the width of the distribution. So you get, and I think this median is just, just the most intuitive thing to use as the typical value for gene expression. And you can ask, well, does it really matter? How much of a difference is there between these values, the mean and the median? Well, we are in a position to calculate it from all this data we just took. And it turns out the errors you make by confusing median and mean end up being around 25 or 30% in the largest cases. So we've got a sense for what that is now. And in some cases that might matter, in some cases it might not. And so, but at least we know what it is. It's well defined now. Okay, so I think we've got, yeah, go ahead.
So usually when you say typical, it's not well defined.
Exactly.
You should say typical with respect to what? What kind of questions you ask?
Yeah.
So your mu is typical for what kind of questions? So
I agree it's a semantic issue, and people are often just blindly assuming that mean captures the typical value, the value of a typical cell at some level, in a way that's not well defined. And the point of this argument is that there's more than one way to characterize a distribution. They're not always the same number, and you need to think carefully about it in cases where it's not just normal statistics. And I think this is a good example of that. I think you can make cases mean, median. They're useful in different scenarios, but you need to be aware of why you're using one versus the other. Yeah?
So going back to the genes that are bimodal and expressing this, did you ever look into maybe if it's possible to look at just one mode of the two and see if that also fit a gamma or a log-normal distribution better? Or I guess I'm wondering, with those binomial or bimodal expressing cells, is it that there's two different gamma functions going at the same time? And they're
Oh, do we fit them to a sum of Gaussians or sum of gamma functions? And that's a good question. I don't recall if we did that or not. They certainly don't fit to one.
Because it seems like
you get enough parameters, of course, you can fit an elephant, make its tail wiggle, and all that. Yeah. Okay. All right. So now that we have some understanding of the noise, we can turn to questions of, what's the systematic variation between cells? And we've kind of embarked with our collaborators on this program of trying to reverse engineer complex tissues. So taking something like a colon or a breast and trying to dissect all the different cell types that are there within it, from sort of stem cells to fully differentiated cells. And this is something that has been worked out most gloriously in the blood over the past couple decades. There's a sort of a reasonable consensus of what the hierarchy of different cell types are in the blood, starting from the hematopoietic stem cells, which can make the whole blood, and they go through a series of differentiation steps to sort of become more specialized cells and turn into B cells and T cells and everything else. It's part of the blood and the immune system. And we want to kind of do that for solid tissues. And people have been trying that over the years, and some of the popular tools have been flow cytometry, which works great for things like blood, and have now been more recently applied to solid tissues. People dissociate the cells and put them through the cell sorter. Immunohistochemistry, where you stain things with antibodies, look under the microscope, is another powerful tool. And we think these single-cell gene expression measurements add a very nice complement to that by letting you look at many more genes than you could in these other methods within the same cell. And by having the ability to easily design primers, you can sort of look at arbitrary sets of genes. You're not dependent on the ability to make antibodies. And so we're basically combining all these together to try to dissect these cellular hierarchies. And everything I'm going to describe in the next few slides on colon has been done in collaboration with Mike Clark. And the basic experiment is to take the primary tissue, in this case a bit of colon, dissociate the cells. So now instead of being all glued together in one big mass, they're all separated. We can take that single cell suspension, label them with antibodies if we want to look at different surface markers, put them through a fax machine, so separate them out according to which surface markers they have. And then the fax machine lets you drop single cells into different single wells in a 96 well plate. And within those wells, the buffer makes the cells lyse and preserves the mRNA. And then we do a preamp PCR within the wells, which lets us sort of boost up the RNA levels. Then we load them into the Fluidigm chip and do the measurement. Okay? And this has actually been the procedure, this sort of single cell fax preamp and then measuring the chip for everything I've described in the noise section as well. Okay? And all those cell line things were done with this process. And we've shown that all the noise measurements include the effects of this preamp, which is actually quite low. And so then at the end of the day, we get these interesting these patterns of gene expression, which I'll describe in more detail. Is the experiment sort of clear here? Yeah.
How much do you know about how the gene expression level changes from going from tissue to sorting? So that's a certain amount of time when they're dissociated.
Yeah, that's a really good question. It comes up not just for our measurements, but anytime you're doing this. Came up for the blood guys, comes up for any other sort of fax-based measurement. And at the end of the day, what you sort of go back to is sort of functional measurements. So you show that you can take these cells after they've been sorted and purified, implanted back into mice, and they retain some of the function that you were interested in. So in the case of blood, you show that you can take the purify hematopoietic stem cells, implant them into a lethally irradiated mouse, create a new immune system. So the sorting hasn't destroyed the ability of the cells to do the function that you're interested in. Same is true for cancer stem cells. You sort them from a colon, from a human colon tumor or breast tumor, pull out the ones that are tumorigenic, plant them in a mouse, make a xenograft, they can create a tumor. So they retain their function. And that's sort of an important piece of it. And so from that accumulated evidence, people have a sense that you're not doing too much distortion of the cells through this process. Or not doing beyond repair, let's put it that way. Yeah?
How do you get the right figure, this expression? From after the sorting, you get all the different cells in the different columns. Then how did you get the expressions?
How do we get the expression?
Yeah,
So,
from this middle step to the right step.
okay, so cells are dropped into the wells in the 96 well plate.
Yeah.
They lyse there. We do a little preamp PCR to boost the cDNA levels. We load them into the chip. So the chip gets 96 cells, 96 primer sets, and does 10,000 real-time PCR measurements. And then that data is clustered together to make figures like this, which I'll describe in a little more detail in a couple slides. Okay, so colon was something we started with. It's sort of a nice one to warm up with because there's an interesting connection between geometry and cell fate decision. So your colon is made up of these so-called crypts of Lieberkühn, and at the bottom of the crypt sit the stem cells, and they divide and throw off progeny, and these progeny sort of move up towards the top of the crypt. And as they do, they become ever more specialized in their function until they reach the top of the crypt, and they've specialized into becoming either goblet cells, which secrete mucus, or Pterosites, which absorb things. Okay? Those are the two fates. And then they get sloughed off at the top, and then the whole process repeats itself, and it does this basically every week. So this is a huge amount of turnover going on in your body all the time. Your colon is constantly creating these cells and then sloughing them off. And some of the biology around these have known it's a very well-studied system, so that's good because the geometry is associated with the fate. That makes it easier to interpret in immunohistochemistry. And there's surface markers that will stain the top of the crypt, the bottom of the crypt, and we can use that to kind of double-check all of our experiments. Okay? So my postdoc, Tomer Kaliski, who I introduced you to before, and Mike's postdoc, Pierre de Lerba, did a bunch of experiments on mouse crypts where they would take the crypt of a healthy mouse, dissociate it, do everything I described, did the gene expression measurements, Here's the genes on this axis. So each row here is a different single cell, and each column is a different gene. And the gene expression patterns have all been clustered together. And so you might be very familiar with these sorts of diagrams if you've looked at gene expression data from lots of patient or tumor samples and whatnot, where every column is a gene, every row is a different patient sample. Here, every row is a different single cell from the same mouse. Right? And we've clustered them together according to their gene expression patterns. We only use the RNA measurements to do the clustering. And remember, I told you there were surface markers we could use to mark where the cells came from, so we could retain some knowledge of their geometry. We didn't use that in the clustering at all, but we can use that now to see, does the clustering bear any resemblance to reality? And it turns out, yes, the clustering actually does a really good job of separating cells that we know came from the top and were more mature from the ones at the bottom, which we know were stem-like. So at the grossest level, it all hangs together. But you can see that there's also some structure on here that goes beyond what you can resolve with these surface markers, and you can kind of look at it by eye and start drawing lines that separate out different sorts of putative cell types. And we've given them names here. Okay? And we've given them names because of the particular genes that are expressed in the different groups. This one right here we're calling Pterosites because this gene right here is the gene that defines enterosite function, right? SLC26A3. It's involved intimately in the absorption. So if a cell is expressing that, it's an enterosite. And you can see that it's basically not expressed elsewhere. Okay? So we call those enterosites. Likewise, we call these goblets right here because they're the ones that are expressing mucin. This is the gene that makes mucus, the lubricating gel. That's only expressed in this group. These must be the differentiated goblets. Right? Pretty straightforward. Now, if you look in the goblets, we actually try to divide them into two subpopulations because of this group of genes that's expressed largely in this one. And these are ones that are related to sort of stemness and sort of immaturity. And any of these genes, if you're interested in developmental biology, these are ones that are you associate with stem cells. And so it seems like this might be a sort of population of immature goblet cells, or ones that haven't fully differentiated, and they haven't sort of become fully assuming of their fate. So Pierre went back and did immunohistochemistry to check that, and lo and behold, he found a bunch of cells that were expressing mucin and knob at the base of the crypt, and was able to verify this was a new sort of subset of cell types that came out of these PCR measurements, these single cell measurements. So, and then how about the ones that weren't differentiated into enterosites or goblets? Well, those we think are stem cells because they're expressing a bunch of genes related to stemness, and there's two clearly separate populations. And it turns out it maps very nicely onto a big debate that was going on in the literature. Hans Clevers was a very strong proponent of the idea that LGR5 was the key gene for stem cells in the colon, and Mario Capecchi was saying, well, BMI1 is the key gene that's expressed by stem cells in the colon, and it turns out they're both right. So we have two stem cell populations, one of which is expressing LGR5, another one's expressing BMI1, and they appear to be fairly distinct. And one way in which they're distinct is that the LGR5 ones are having a much higher expression of genes involved in cell cycle and division. And so it appears that these are ones that are actively dividing, these are ones that are quiescent and are waiting there at the bottom of the crypt for when they're needed. So a bunch of cool insights about stem cell biology have come out of these. We published quite a bit of this data on the colon in Nature Biotechnology in 2011, but not all of it. And I'll just say that once you've started to go down this path of trying to find interesting genes that are expressed by different cell populations, you can do all kinds of cool things and pick out a bunch of genes that are related to self-renewal, to cancer, things that might be drug targets in tumors. And we've gone on and done similar experiments in tumors of the colon, both in mouse and in human, and have been able to see interesting subpopulations, interesting potential drug targets, and that's led to a new company that Mike and I founded called QuantaCell Pharmaceuticals, which is trying to develop small molecule therapeutics based on these targets that have come out of these cellular hierarchy measurements.
One question.
Yeah.
How do you interpret the notch pathway? Because you've got like
Hold on, I'll come back here.
this population on the right notch and delta is expressed later. I mean,
Okay.
in a different population.
All right. So there's this one right here, notch, right? It's in one but not the other. You're asking about that?
And but the delta, which is involved in the notch pathway, is expressed in different cell population.
Yeah, because notch and delta, they interact between different cell types anyways, right?
Yeah.
It's so not surprising there might be a different one.
I mean, is there any biology beyond you could share from that?
Oh, undoubtedly there is. And yeah, I don't have a story around that for you right now, but yeah, that's one that Mike's lab is really focused on. They've been trying to work out these pathways and what's going on. But it points out that, these experiments are great at generating hypotheses and putting you on the trail, but you still have to go and do the biology at the end of the day. I wouldn't want to oversell it. So that's all. All of those measurements were based on PCR, which required you to make some decisions ahead of time about what genes you wanted to study. And 100 genes is actually a big number. And I used to tell my collaborators, don't be lazy. Any biologist worth their salt can guess at 100 genes that may be relevant to the problem they're interested in. I'd always say, I want to do an unbiased measurement. I want to do RNA-seq arrays or things like that. So we eventually got dragged into doing that. And this sort of ability to do single-cell RNA-seq is something that has become quite active in the literature in the last year or two. A number of papers from different groups around the world showing how to do it, both in microliter volumes and also Fluidigm has developed a chip to do it in nanoliter volumes. And so we've been kind of playing around with both and decided to sort of compare them all head to head and see what the performance was and use our sort of finely tuned knowledge of sort of measurement precision from the PCR measurements to try to understand how good the RNA-seq measurements were with these different methods. And this work is actually going to be published, I think, this weekend. It's coming out in Nature Methods. This was, I'll show you the picture on the next slide. But basically, here's an outline of the experiments. Okay, so we did a bunch of tube-based single-cell RNA-seq experiments using published protocols. Looked at this smarter kit from CloneTech and this other one from Sigma with a few cells each. We did a bunch of PCR measurements using the methods I described earlier. And then we did the microfluidic chip called the C1 that Fluidigm has developed, where we've done both PCR and RNA-seq on larger numbers of cells because the automation makes it easy just to crank through huge numbers and do that rather inexpensively. And then everything was sequenced. So RNA-seq, in case you don't know, is a way to use these next-generation sequencers to measure gene expression by essentially sequencing all the RNA molecules in a sample, mapping them back to the genome and inferring gene expression. And this work was led by Angela Wu, who's a postdoc in the lab. And so, it's fairly reproducible. If you ask, I should also say we also did experiments on bulk samples as kind of our references. So if you ask, how many genes are repeatably detected in multiple replicates, in the bulk samples is pretty good. In the single cell samples, it's lower, but reasonable, and a little better in the microfluidic one than the others, but they're all sort of comparable. And that is what it is. You can ask about the measurement levels, how good is the quantitation, and we tested that by spiking in controls into each single cell experiment. And so there's a NIST traceable RNA standard called the ERCC standard, which you can spike in, that you can use to calibrate any sort of RNA measurement you want to make. It's a large number of transcripts of various sizes, complexity, and levels, and it's sort of defined down to the number of molecules in it. So that's a nice thing to do. We put that in. This is the data from the microfluidic experiment. This is average expression levels over 96 replicates in that chip, and you can see that you're getting very sort of linear expression. And so this is now log on this axis, on both axes actually, and you can see it 1 molecule per chamber. You've got pretty good sensitivity, and that's great. And that corresponds to an FPKM of about 1, which is, in technical terms, that's about what you'd expect for this value. So in absolute values is pretty good. And you can see that we're actually measuring things that are present at a value less than 1 molecule per chamber. How do you do that? Well, remember, we are averaging across 96 chambers. And so if you have something that's on average a tenth of a molecule per chamber, that means you'll see it in 9 chambers or 10 chambers, and the rest will be empty. And on that kind of averaging across the chambers, you're able to preserve dynamic range. That's pretty good. We're happy with that. The other thing that was pretty neat to see is that the number of transcripts in a given cell is not that high, about 250,000 or so. And so you'd expect that you wouldn't have to sequence very deeply to sort of saturate all the genes. And that's indeed what we find. And so in this single cell experiments, that's the bottom 3 curves right here. This is how number of reads sampled, sort of a rarefaction curve. The deeper you sequence in, the more genes you find. This is the number of genes you detect. And you can see it increases dramatically, and then it saturates, okay, right around 500,000. And the one that's finding more genes is the microfluidic one. It was doing a little bit better. These are the bulk measurements right here, the red and the yellow. So you can see that, the 2 different ways we use to prepare bulk samples gives you different numbers of genes. So it's an interesting thing to keep in the back of your mind, that there are systematic things that come out of the particular enzymatic scheme you use to make and amplify cDNA. This is the one that corresponds, this orange one is the one that corresponded to the method we use for the single cell. So you can ask, if I take all these 96 single cell measurements and combine them together synthetically, by combining the data together, and I pretend that was a bulk experiment, kind of a synthetic ensemble, what does it look like? Well, that's the green curve right here, okay? And so it actually, the collection of single cells does seem to reproduce the bulk ensemble in a way that's pretty good. And this is just now in kind of number of genes detected. You can ask the same question in terms of expression levels, and you see something like that here. And so the synthetic ensemble recapitulates the bulk. And so you can say it's sort of sufficient to do a bunch of single cell experiments, and I'm getting the same answer I would if I was doing the bulk. And all that extra information that comes from knowing which genes were expressed in which cells. Yeah. I mentioned also that we wanted to sort of compare absolute expression levels, really measure accuracy of RNA-seq measurements, because we worked so hard on PCR and we think we really understand what we're measuring there. And this now is what you get when you sort of compare all that together. So each panel here is a different gene. The left one is the RNA-seq, the right one is the PCR, and these are the expression value distributions, and they call these violin plots, because they've just taken the distribution, turned it sideways, and reflected it about its axis. So you really want to think about it like, all right, turn your head sideways and look like that, and it's a frequency distribution. And don't worry about the absolute values here. They're all kind of offset in funny ways. But look at the widths of the distributions, okay? Because that tells you sort of something about this interplay between technical noise and stochastic noise and intrinsic biological noise. And for things like beta-actin, they actually look pretty comparable. Remember, we're pretty confident in the PCR measurements, and the RNA-seq looks like the PCR. Oh, that's good. And GAPDH also looks pretty comparable. But then you can find other genes where the RNA-seq measurements are much broader and the technical noise is much higher. And so I think with the RNA-seq, we're not quite all the way there where we're limited only by stochastic noise. We do have technical noise for many of the genes that is limiting the measurements. And so for measuring distributions of noise, you've got to bear that in mind. That doesn't mean you can't find a good central value of the distribution or a good mean expression level, but sort of on a per cell basis, you've got to realize that in many of these genes, RNA-seq is going to be putting on some technical noise. And then we can look at the mean expression levels by both methods. Here's 3 different ways of doing the single cell RNA-seq. This is the microfluidic one. These 2 are microliter ones. And so this axis is each point is a gene averaged across all the cells we measured. This axis is expression level by PCR. This axis is expression level by RNA-seq. And we can ask, do you get the same answer with two different methods of doing gene expression? And it turns out it's actually not that bad. The slope is 1. It was amazingly good. And even if you take these outliers out and just look at this part, you still get a very good slope and correlation. It really, quite impressed. And so I feel like you can get reliable values out of these RNA-seq measurements. They do bear some connection to reality. It's not all been distorted by the huge amplification. Yeah?
The previous slide, there was some distribution that essentially went from a single mode to 2 modes. Is this something real? I mean, for example,
pick one.
On the right there, there's
this one.
So it looks almost like as if it switched from a single mode to 2 modes. And how would noise introduce a bimodal distribution?
Yeah, it's a good question. It could have to do with GC bias and things like that. That things are getting differentially amplified by the PCR. There's all kinds of subtleties that come into that. And when you sequence, everything comes out the other side. And so you could be looking at sort of bimodal amplification efficiencies for different parts of the transcript, for example. Okay, I think the last thing I'll tell you about here is just a couple of slides about how we've been using this single-cell RNA-seq now to try to go look at another tissue in mouse, this time the lung, and see if we can't do this reverse tissue engineering with the full RNA-seq information. And this is work that's been done by a postdoc of mine at lab named Barbara Treutlein, and in collaboration with Mark Krasnow's group and his student Doug Brownfield. So the basic experiment, here this data is E18, so very early mouse embryo. The lung is dissected, the cells are dissociated, purified by Max, and then put into which is a bead pull-down scheme, and then put into the Fluidigm chip where we do the single-cell transcriptome amplifications, and we sequence it and read out the gene expression. Okay, so the alveoli of the lungs, which is the part we're interested in looking at, has a bunch of different cell types where, again, people have been kind of cataloging them by traditional methods that I described earlier. The 2 major types are called very creatively type 1 and type 2. And they've been associated with certain genes. And so these are sort of the canonical genes that everyone believes mark these cell types. There's the AT1 is sort of the very flat one that creates this big surface area, and AT2 is the cuboids that sit around and interesting things there. And some are ciliated, some are clara cells. These are all ones that are associated with sort of different morphologies and genes that have been measured by staining. So here Barbara looked at what the sort of, she did the experiment, did the RNA-seq, and then looked at what happens when you just cluster together by the known marker genes and ask, do you start to recapitulate patterns that make sense and agree with what everyone believes? And the answer is more or less yes. The right genes are clustering together in the right groups. If you do PCA analysis on the whole, principal component analysis on the whole set of genes, and then ask how are they loaded by the different marker genes, they tend to all point in different directions, and the same colors, which are the same markers for the same types, tend to be pointing in the same direction. So it all kind of deconvolves the way you hope it would and sort of matches up with known knowledge. And here's the first 2 components, and you can see that as we call the cells, they are clustering together in principal component space in the way you'd like. That's sort of a reasonable sort of check that it's all going the way you hope it would. Now you can ask, what other genes are associating with these different cell types that are popping out of the RNA-seq that you wouldn't have known about ahead of time? And so Barbara started looking for things that were correlated with these different marker genes and found some really interesting things that are creating really interesting hypotheses about what might be the lineages of development and what are the sort of the key things going on in different cell types. And so especially things like HOPX is one that we've been following up on quite a bit. We've gone back and done Doug has gone and done a bunch of immunohistochemistry, and so, I'm blanking on the word now, fluorescent in situ hybridization, to validate a bunch of these, and they're all coming out the way the RNA-seq said they would. So it's given us a whole set of genes to explore in the development of the mouse lung that is going to keep us busy for a bunch of years to come. I think I'll stop there rather than get into another story and cram it all into 5 minutes. And I've tried to acknowledge the folks who've done the work as we've gone through. I'm happy to take any more questions you might have about this work. Thank you.
I've got a question. So when you look at the correlation between protein expression and messenger RNA level, you looked at EGFP. What promoter did you use?
What was the promoter? It's a constitutive promoter. I'm blanking on the name right now. It's one of the standard ones. But it's in the paper. That part's published.
And do you expect, or have you seen different, not as strong correlation with different promoters?
If you vary the promoter, would you see I would imagine you'd see variations in correlation. Yeah. This EGFP is kind of a gimme example because, you put it endogenously, you'd expect those to be very correlated. Maybe it's expressed at a non-physiological level. But that's why we looked at the surface markers too. And so, right. So we looked at the surface markers, which weren't engineered in any artificial way and not correlated as well.
Yeah. So you showed the single cell level, you had about 3,000 RNAs, but then when you put them together, you had a total of 9,000 possible RNAs being. So that seems like an awful lot of noise at that level, and presumably that's resolved. You have these things being translated into protein, and somehow that has to be dealt with because some of these things might be inappropriate or something for the cell. So do you think that sort of robustness is happening more at the protein-protein interaction, and more noise has been allowed to evolve at the RNA level?
Yeah, that's a really intriguing question that always comes up. Sure, the RNA is noisy, but there are other methods of control that are more tightly regulating protein expression. And it always seemed like a nice escape hatch. But Johan Paulsen and his collaborators have been publishing some really nice papers about how much can you squeeze the noise down using arbitrary control techniques. So he's been using the tools of control theory to say if you had some arbitrary complicated control network, how well could you do with squeezing the noise under very, very general assumptions. And under these fairly general assumptions, the answer is not very well at all. So it's very hard to squeeze the noise. It's a head scratcher.
So now that you can actually get a gene expression fingerprint from particular cells, one can probably ask how this correlates with actual phenotype. So, for example, I take two cells which naively I would expect to have similar gene expression pattern. Are they really indistinguishable to the level of to the degree you can measure them? So can you take particular type of cells and see how much variation is there?
Yeah, that was sort of the point of these stochastic noise measurements, where we were, for almost all those experiments I described, working with clonal cell lines. They should all be exactly the same. They're like twins.
Okay.
Yeah. So it doesn't get more alike than that.
Then you say this noise, is it really coming from your measurement technique, or it's real?
So for the case of these PCR measurements, we believe that our measurement noise is substantially lower than the biological variation between the cells. And we believe that the biological variation we observe is consistent with these models for stochastic noise and gene expression that I'm putting up there.
It may also depend on the time, right? So you measure a particular moment of time, and presumably the gene expression level is time dependent, right?
Depends on the gene. So, you know, so-called housekeeping genes tend to be expressed fairly uniformly through the cell cycle. Other ones are going to change in time. When we sample the cells, they haven't been synchronized anyway, so we're sampling over all time points essentially by doing these, you know, several hundred replicates in each experiment.
Any other questions? Let's thank Steve Quick.